So, hello everyone and welcome to this video. In this one I would like to demonstrate to you a function from R and also a quick workflow that allows you to extract these numbers that you can see here in the workspace. This is actually a very common procedure. After we have conducted or decided for a gating strategy that we now trust in, we usually want to reduce the big amount of information to only a few numbers that we can use for plotting or for applying statistical tests. And these are either cell counts or percentages or calculated statistics like MFIs. The focus of this video really is to demonstrate the technical aspects and not so much the biology. Still, of course, I want to tell you what kind of data we are looking at here. These data come from a retroviral transduction where a GFP gene was transduced into epithelial cells. And the question is, the variation among these 48 files that we have here in our workspace, which of these variations are most suited or best to induce a high frequency of GFP positive events in the end. So that means which condition is best to make the transduction efficient. I have already conducted the gating. You can see here that I'm gating for epithelial cells in the scatter, for W negative events, and then for the very bright GFP expressors. The important thing to know or to just recognize is that these workspace files, they are actually, or they come in the XML format. So a very hierarchical file format. You can see here it is an XML. And many information are really hard-coded and saved to disk. For example, if we scroll down here, we can see that all these keywords from our FCS files are saved within this file. And moreover, I can also see that counts appear here, like this one or that one. And it is possible to extract these counts independent of the FCS files. And this is what I'm going to show you with the function in R. Right, so we are going to use a package which is called fcexpr, and it is available on GitHub. You can just install it by using these commands. I would just skip that because I already installed them, so I will just start with these library commands that attach the packages for the current R session. And the function that I'm talking about, or that I want to demonstrate, is called WSX get popstats. So WS stands for workspace, and X refers to the fact that we are reading this XML file. As usual, if you want to know how this function has to be used, you can just type a question mark and then the name of the function and hit enter. And then you will see a quick description. You will see what kind of arguments are required to call this function, and you also can see what you get as a return value. And the only thing that we have to provide here is WS, which should be the path to a FlowJo workspace. So let's just do it. We go to the Finder, copy the path of this workspace file, and paste it here. And then we execute the function. And within seconds we get the return, which is a list with 2 entries. And these 2 entries are counts and stats. Now, continuing with counts or stats is very much the same principle, so I will just focus on the counts. If we want to access those, we can do it like that. Now we get a table, or a data frame as we call it in R, and this data frame contains 4 rows for every of our FCS files that we saw in the workspace. And these 4 rows correspond to the 4 gated populations, including the root itself. And for every of these populations we have the counts, the parent count, and from these 2 the calculated fraction of parent, or frequency within the parent gate. And this is what we want to visualize in order to answer the question, which experimental condition or setting is best to maximize the frequency of GFP positive events. This plotting or analysis could be done in R, which has a few benefits, but it can also be done in Excel, as most of you will know it. So if we want to save this this table to Excel, it can be done very simple with a function from another package. We just have to say what we want to save. This is PS popstats, the table that we have here, and then we have to say where we want to save it to. So we just do that, hit enter, and then this file appears here on disk. It's now the same table that we saw in R. So and now we could, for instance, filter only for the GFP positive ones here, and then copy the frequency of parent and plot, or make a test, or t-test, or whatever. But the thing is, no matter how we continue, if we use Excel or if we use R, what we need now in order to visualize the results is we need so-called metadata. So it means we need a table that tells us what these files actually represent. And I already have prepared such a table here. It's called a sample description. We have one row for every FCS file, and then we can see what has been varied in this FCS file or for that FCS file. So here we can see that variation has been done with respect to the viral supernatant volume applied to the cells. Variation has been done. The incubation time of the viral supernatant on the cells has been varied, as well as the percentage of fetal calf serum and the seeding density. And we want to know which of these factors is most influential or has the biggest impact on inducing a high frequency of GFP positive events. And seeding density actually means that I have seeded the cells in a very dense fashion or in a very sparse fashion. So 16x is the highest dilution. If we don't have such a table ready, we could initiate it using the help of R. Here we have a function which is called list files. We can give it a path and we can say what kind of files we are looking for. So we are looking for files that have FCS in their name, which are our FCS files. If we execute that, we get a vector with all these paths, and from that we can also extract the file names, like that. Then we could make that a data frame. We have it here now, data frame with these two columns, and this can be saved to disk then, for instance. So again, we just say what we want to save and where we want to save it. My metadata, and this could be now the starting point to create such a table. So but we already have that. So let's come to the actual pipeline that allows us to quickly read the data, connect them with metadata, and then plot. So we would, if we start over now again, we would just read the whole counts or the whole statistics from the FlowJo workspace. Then we would focus or filter on the GFP positive population only. So this is what we do. And then we would read the sample description also into a table. It looks like this. And you may notice now that in the sample description we have a column which is called file name, and the same column also exists here in our data frame with counts and frequency of parents. And these matching columns can now be used to apply one of these powerful joining functions that will combine these two tables into one. And here we just focus on a few columns that are relevant. And then you can see that very quickly we have combined the phenotype data, or what has been analyzed, with the metadata. And this is the same principle always, yeah, in order to plot or analyze the data by statistics. So this data frame is ready now for plotting, and we will use functions from the ggplot framework, which also belongs to the tidyverse. We will just plot one of the variables as the x-axis and plot the frequency of GP positive events on the y-axis. And if we do that for the virus supernatant, we can see that there is a slight positive effect. That means if we use more virus supernatant, we get a higher frequency of GP positive events. The same is true for incubation time. If we increase it, it seems to be beneficial with respect to inducing a high frequency of GP positive events. So 48 hours seems to have shifted up a bit in comparison to only 2 hours incubation time. Using an increased amount of fetal calf serum, at least when we compare these two factor levels, does not seem to be does not seem to have a big impact, whereas using a very sparse seeding density seems to really have a large impact on the frequency of GP positive events in the end. And this could totally make sense, because retroviruses, they need proliferating cells in order to infect them. And if we seed the epithelia very sparsely only, they have more room to expand and they will probably have a higher proliferation rate. And since that, we see a higher rate of infection with the virus, which is indicated by the expression of GFP. So here we have visualized every one of these 4 factors one by one. But with a little bit more practice and a little bit more experience, it is also very simple to visualize 2 factors at a time with the response as a color scale, or even it is possible to very quickly visualize the whole complexity of our experiment with only one command in the end. So here we have seeding density, incubation time, and here in these so-called facets we see the variation between percentage of FCS or the amount of virus supernatant. And in that way we have a tool or a pipeline that allows us to very quickly visualize the result of such an experiment. And now imagine the following situation. You show these results to your group, and then your supervisor says, yeah, very nice, but please make this gate a bit more like that. I don't like the other one. And you are now, with the help of this pipeline in R, you are able to respond super quickly to such a request. You just change the gate, apply it to the group, and save the workspace to disk. Then you go back to R, just execute these few commands, and within seconds you get an updated plot that tells you how the change of the gating impacts the final result. So this is before with the old gate, and this is now with the new gate.